Good morning, students. Today we are going to learn the topic human health and disease. Already we studied what is meant by immunity. Now we are studying about the acquired immunity. First of all, we should know what is meant by acquired immunity. Acquired immunity we are getting after the birth. So here you can see the definition of that. Ability of the body to define itself against invading foreign antigens. Ability of the body to defend itself against invading foreign agents. Foreign agents in the sense bacteria, virus, protozoa, fungus, etc. Those things are we call as foreign agents. So when these foreign agents enter into the body, our immune system immediately reacts. This pathogen, that means acquired pathogen, this pathogen is a specific immunity, pathogen specific immunity. Acquired immunity, it can identify the pathogen, so it is called as pathogen specific immunity. It's already I told you, they developed during the lifetime. It's characterized by memory. It is characterized by memory. In this case, once the disease occurs, in that pathogen automatically it hold in this memory. So once the next time if it goes, automatically it affect the antigen. So that we are saying that is characterized by memory. By a body encounters a pathogen first time is called a primary response. See, for example, the disease measles attack us. Once it comes in the first time, that is our immune system to recognize. It takes time to identify the particular pathogen. So once if it is recognized, then automatically affected. In that response is called as primary response. So the primary response, this is the first time. Now it hold in its memory. Next, what happened? The second encounters of the same pathogen. See, for example, bacteria. Bacteria means the same bacteria enters again. Bacterial disease. Again the same bacteria enters. Immediately our immune recognize the same pathogen. So the function is very fast. The high intensity is high. That's why it is called as memory. Acquired immunity is having the memory power. So students, we studied about what is meant by acquired immunity. There are two responses we study, primary responses and secondary responses. Apart from that, what are the features of acquired immunity? There are 4 different types of features. How many types? There are 4 different types are there. What are they? Specificity, diversity, discrimination, and memory. What are they? Specificity, diversity, discrimination, and memory. First, specificity. Specificity, ability to differentiate among various foreign molecules. That already we studied what are the foreign molecules, pathogens, different kinds of pathogens. So it can easy to specify which pathogen enters. The next for the diversity. Diversity in the sense it recognizes the vast variety of foreign molecule group. How much numbers of pathogens are there? The vast numbers, that means enormous numbers. Among that numbers also it can easy to identify which pathogen it is. The next for the discrimination, ability to distinguish between self and non-self. See, suppose for example, organ transplantation takes place. During the organ transplantation, we are receiving the organ from the donors. So it should recognize whether it is self or non-self. So that is comes under the discrimination. The next for the memory. Memory, already we studied this character, this important character of this acquired immunity is the memory, retains memory of previous infection. Then second infection with the same pathogen brings quicker and stronger immune response. This quicker and stronger immune response is otherwise called anamnestic response. We will say Anamnestic response. ANAMNESTIC. Anamnestic response. These are the features of acquired immunity. So students, what are the different features? Specificity, diversity, discrimination and memory. Now we can study about the immune response. Children, we studied that there are two types of immune responses. What are they we studied? Can you say the answer? Primary response and secondary response. So this primary response already studied. The memory is low. So low intensity. But the secondary response, high intensity. Now, this primary response and secondary responses are brought about by lymphocytes. Okay. Now we can study about the lymphocytes. What are the different types of lymphocytes? What are the differences? There are two types of lymphocytes. What are they? B lymphocytes and T lymphocytes. B lymphocytes and T lymphocytes. The B lymphocytes, its function is an body fluids. Blood and body fluids. It is important for the formation of antibodies. Formation of antibodies. Now, this T lymphocytes, it does not involve for the formation of antibodies. At the same time, it is important for the organ transplantation. Important for the organ transplantation. So, these two things we are studying about the lymphocyte. There are two kinds of lymphocytes, B lymphocytes and the T lymphocytes. B lymphocytes functions is in the blood as well as in the body fluids. So, it is involved for the formation of antibodies. Then for the T lymphocytes, it does not involve for the formation of antibodies. At the same time, it stimulates, that means it is support to the B lymphocyte to the formation of antibodies. The special feature of the T lymphocytes is it is involved for the organ transplantation, for tissue matching. This T lymphocytes involved for that. So for the cells and organ, for those functions is performing. So this T lymphocytes otherwise called cell mediated immunity. D will come, cell mediated immunity. This B lymphocytes otherwise called humoral immunity. Humoral immune response. These are the things we are talking about the lymphocytes. Now, we can learn about the structure of the antibody. Structure of antibody molecules. Here in the board you can see the image. Antibody molecules. By seeing this one, which letter is, remember children? There is the Y letter, right? So, this antibody, the antibody is, what shape it is? Y-shaped. It is Y-shaped structure. Y-shaped structure. Here you can see 1, 2 small line and 2 long lines are there. 4 lines are there. So this antibody has 4 peptide bond. Peptide bond. 4 peptide bond, otherwise called peptide chain, we can say. Peptide chain. Peptide chain. Now you can see here there is the 2 short chains are there. This one and this one, 2. These 2 short chains are called light chains. 2 light chain, 2 light chain, and the long lengthy one here. This one, second one, sorry, this one and this one. So 2 long chain also is there. So in both the cases, this also comes under the called a heavy chain. Is called a heavy chain. So antibody, what is the structure? It's a Y-shaped structure. 4 peptide chains are present. There it is. Consists of 2 light chain and 2 heavy chain. Now you can see there is the bond, right? So here this in the bond is present. There, that is consists of sulfur. So this one disulfide bond. How many disulfide bonds are present? We can count it. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So there are 16 disulfide bonds are there. Disulfide bonds are present. Then in both sides, here where the light chain and the heavy chain, there you can see there is a room-like structure. That means lock and key structure. This structure is advantage to the binding of antigen. So this site is called antigen binding site. Is called antigen binding site. Binding site. Children, listen, it's very important for this diagram. For a case-based question, you can expect this question. Okay, nothing but here there are 2 chain, 2 heavy chain and 2 light chains. So totally 4 chains are there. That's why it is called as 4 peptide chains. Then Y-shaped structure. Then both the end of the light chain and the heavy chain, there is a special area is called as antigen binding site. So when the pathogen enters there, immediately antibody engulf it. The next it has the sulfur disulfide bond. Totally how many, if it is asked, even the NEET question also is important question. 16 disulfide bonds are present in the antibody. So children, today's class, what are the things we studied? What is meant by acquired immunity? Specification, special features of acquired immunity, the human responses we studied. Now we studied about the antibody molecules. Okay children, we can see in the next class. Thank you.